Hello students, welcome back to the channel. In this video, we are going to study digestion in small intestine. See, in the previous video, we have already covered how the digestion was occurring in the mouth and in the stomach. This video, we are focusing on the digestion in the small intestine. So see carefully, various type of movements are seen in the small intestine, which are because of the muscular movement. Muscularis layer is showing lot of movements in the small intestine. It's a type of a mechanical digestion. Okay, and these movements will help in the properly mixing of the food with various secretions so that the digestion can be done properly. Now, what are the secretions which are coming in the small intestine? The bile coming from the liver, pancreatic juice coming from the pancreas, and intestinal mucosa is secreting the intestinal juice. These are the three secretions which are released into the small intestine. Clear, beta? Now I'll show you the diagram. Now see, this is a type of a segmentation. Segmentation is occurring in the small intestine. It is a type of a movement in which ring-like constriction is going on along the entire length of the small intestine. So that you can see in this diagram, suppose this area is in the, where the food is there, then is going for the segmentation, and see carefully the mixing is going on. Okay, simultaneously contraction and relaxation is going on. As a result, properly mixing of the food is done. So this is a type of a movement which we study in the small intestine. Now see next, if you see the liver, see this gallbladder and the pancreas, they are pouring their secretion into the duodenum through the common hepatopancreatic duct. The common hepatopancreatic duct is the one which is bringing the secretion from the pancreas, and it is bringing the secretion from the liver. Another important you try to focus here, duodenum mucosa is also secreting the secretion. Clear, beta? You can see this color, check carefully. Duodenum mucosa is also secreting the secretion here, that is intestinal juice, and that is known as succus entericus. So hepatopancreatic duct is the one which is bringing the secretion from the liver and pancreas, and intestinal duodenum mucosa is secreting the intestinal juice, collectively known as succus entericus. Now see, what are the secretions which are present in the pancreatic juice? What are the things present? Now see this diagram carefully. See, pancreas, this is pancreatic juice, and it contains three inactive proteolytic enzymes. One is trypsinogen, second is chymotrypsinogen, and third is procarboxypeptidase. These three are proteolytic enzymes which are made in inactive form, present in the pancreatic juice. Next, they contain the carbohydrate digesting enzyme pancreatic amylase. Breakdown of fat is achieved with the help of pancreatic lipase. Next is digestion of the nucleic acid is with the help of nucleases. So pancreas is releasing the enzyme to digest all biomolecule. For protein, proteolytic enzyme. For carbohydrate, it is amylase. For lipid, it is lipase. And for nucleic acid, it is nucleases. Now you have to focus here carefully. Try to focus in this line, beta. Trypsinogen is inactive form, is activated with the help of an enzyme that is known as enterokinase. Now, enterokinase is the one which is made by duodenum mucosa. See where I'm putting the color, please, you have to focus that color. This duodenum mucosa means coming from the intestinal juice. Intestinal juice is producing enterokinase. This enterokinase is activating trypsinogen into trypsin. Okay, it is activating trypsinogen into trypsin, and this trypsin will then activate chymotrypsin and procarboxypeptidase. You can see here, this trypsin is working as an enzyme to activate the chymotrypsinogen into chymotrypsin and procarboxypeptidase into carboxypeptidase. So it means these are the active form. Which are the active form? Active form is trypsin, active form is chymotrypsin, and carboxypeptidase. Now, what the trypsin and chymotrypsin will do, they will break down the protein into large peptides. And carboxypeptidase will convert these large peptides into dipeptides and amino acids. You can see in the equation in the NCERT also. This is NCERT paragraph. Protein, proteose, and peptones. Proteose and peptones are partially digested protein which were present in the chyme. Chyme is the one which is received from the stomach to the duodenum. Reaching the intestine is acted by proteolytic enzyme of pancreatic juice. Which are the proteolytic enzyme? These three: trypsin, chymotrypsin, and carboxypeptidase. They will break down protein or peptones or proteose to dipeptide. Clear, beta? Now you see here, pancreatic amylase. Pancreatic amylase will break down the polysaccharide into disaccharide. Pancreatic lipase. Lipase is the one which is breaking down fat. Fat is actually triglyceride. This is triglyceride. This triglyceride will be broken down into diglyceride and then into monoglyceride. Nucleases are the one which are breaking down nucleic acid into nucleotides and finally into the nucleosides. So this is how the protein and pancreas enzymes are doing the digestion. They are digesting the protein component of the food, then they are digesting the carbohydrate component of the food, fat component, and nucleic acid. So all four biomolecules are digested by the pancreatic enzymes and different names. I hope you have learned this topic. Now let me show you the NCERT line. So see, beta, we have studied this only. Pancreatic juice and bile, they both are released through the hepatopancreatic duct. Clear now? Now what does pancreatic juice contain? So you can see pancreatic juice will contain inactive enzyme. Which inactive enzyme? It is containing trypsinogen, chymotrypsinogen, and procarboxypeptidase. These three are which one, beta? These are proteolytic enzyme. Clear, beta? It contains amylase to break down the carbohydrate. It contains lipase to break down the lipid. It contains nuclease to break down the nucleic acid. So list of the enzyme already I've shown you, but here also NCERT line I'm trying to match you. Now see next. Trypsinogen is activated by the enzyme enterokinase, which is secreted by the intestinal mucosa into active trypsin. Please focus carefully. Who is activating the trypsinogen? Trypsinogen is activated by which enzyme? Enterokinase. And who has secreted this enzyme? Intestinal mucosa. Now, this trypsin, the activate form trypsin, will activate other enzyme in the pancreatic juice. So I think so now this line of NCERT and that paragraph of NCERT is properly clear to you. Now we will focus on the bile juice. Now see, beta, what does bile juice contain? Try to understand. Bile juice contain bile pigments. Which bile pigments? You have bilirubin and biliverdin. So it contains bile pigment. Then number 2, I'm writing it here. You see it contains bile salt. It contains phospholipid lecithin and it contains cholesterol. This is the component of bile juice. Please focus, bile juice does not contain any enzyme. In the bile juice, enzyme is not there. Then what is the role of the bile juice? Bile juice is responsible for emulsification of the fat, and especially with the help of bile salts. Bile salts are the one which are actually helping in emulsification. Now you can see in this diagram, this is one large fat droplet you can see. With the help of bile salts and phospholipid, this is broken down into small droplets. Okay, it is broken down into small droplets you can see. It means emulsification is done. Now this bile salt, another role of bile juice is to activate the enzyme lipase. They activate enzyme lipase to form the micelle. Micelle is nothing, this structure, but you can see this structure. In this structure, if you see, this is, I'll just focus better, this is the bile salt, okay? This is bile salt. So in the bile salts, you can see blue color. I'll just use this one. Try to focus. This blue color is your hydrophilic part, and this inside orange color is your hydrophobic part. Clear, beta? So bile salt has two parts, hydrophilic and hydrophobic. This is hydrophilic part, and remaining you can understand is hydrophobic part. You can see here in this diagram, this is your phospholipid lecithin. Phospholipid lecithin. And in the center, you can see this is the cholesterol. So this is the micelle formation in which you are finding hydrophobic shell is facing outside and hydrophobic core is in the center. And this is the way how your pancreatic lipase is trying to break down fatty acids and releasing the fatty acid and converting the fat into diglyceride and finally into monoglyceride. You can see this is the monoglyceride. Okay, beta. Glycerol with one fatty acid is monoglyceride, and these are the fatty acids are liberated. So this is the function of the bile juice. Now let us see the NCERT. So see, beta, bile which is released into the duodenum contain what? Bile pigment. Which bile pigment? Number one, we are focusing on bile pigment. Bile pigment like bilirubin and biliverdin. Then it contains bile salt. This is number two. Then it contains cholesterol. Please focus, that is important question, cholesterol. And it contains phospholipid. So bile juice contains these four components. Clear, beta? Important point, please highlight, I'm highlighting it. Bile activate the lipase also. That is why in the diagram I've shown you, it activate lipase. What is the role of the bile juice? Bile juice will help in the emulsification of fat. Emulsification means breaking down of fat into very, very small micelles. Is it clear? And I told you they don't contain any enzyme. Bile does not have any enzyme. So in this video, up till here, we have studied the juice which are coming from the pancreas and from the bile. What are the component of the pancreatic juice and what are the component of the bile juice? And what is the role? Is it clear? We have studied it. Pancreatic enzymes will be breaking down all the biomolecules, and bile juice is having no enzyme, but it is responsible for emulsification, and bile salts are present, bile pigments are present, phospholipid and cholesterol. When they do emulsify, they will convert the fat into very, very small micelle-like structure. So this much is clear. Now we will be studying the intestinal juice. Now see, beta, intestinal mucosa epithelium has goblet cells. So remember, innermost layer of alimentary canal is mucosa, which is having epithelium. It is having goblet cell, which will secrete mucus. Secretion of brush-bordered cell of mucosa along with the secretion of the goblet cell means brush-bordered cells. You can see in this diagram, side diagram, you can check it. This is one brush-bordered cells. If you see, it is having, these are the one which are actual protein which is working as an enzyme. So secretion of the brush-bordered cells along with the mucosa which is coming from the goblet cells together will make the intestinal juice that is known as succus entericus. So what are the enzymes which are present in the succus entericus? Please focus the diagram here. See, first of all, enterokinase we have already studied is the one which is going to convert trypsinogen into active trypsin. Clear, beta? Now, NCERT is telling us, you have to see here also, this juice contain a variety of enzyme like disaccharidase, means they are the one which are going to break down the disaccharide. For example, maltose, you can see in the equation down, maltose, lactose, sucrose. These three are the disaccharide, and to break them, we want disaccharidase. For maltose, maltase, lactose, lactase, sucrose, sucrase. So that is why our NCERT is telling us disaccharidase, for example, maltase. Now you come and check in this diagram. If you see, this is the lactase enzyme, which is going to break down the lactose into glucose and galactose. sucrase enzyme, which is going to break down sucrose into glucose and fructose, and maltase enzyme, which is going to break down maltose into 2 molecules of glucose. Then see, it is also containing dipeptidase, which is going to break down into amino acids. So you can see in the diagram, this dipeptidase is your aminopeptidase, because carboxypeptidase is present in the pancreatic juice, and here which dipeptidase is present, this is aminopeptidase is present. Aminopeptidase, which is going to break down. Then it contains lipase also. So lipase is the one which is used here, which is going to break down diglyceride into or monoglyceride into fatty acids and glycerol. So please remember, lipase is present in the gastric juice. Please, I focused in the previous video. Gastric lipase is there, pancreatic lipase is there, and in succus entericus also lipase is there. Though lipase is, fat digestion started only in the small intestine, in the stomach it is not started. Next, it is having an enzyme nucleosidase. So you can see nucleotidase is present and nucleosidase is also present. So you can see here in this diagram, this is nucleosidase is present, and nucleosidase is the one which is acting on the nucleotide to convert them into nucleoside. And nucleosidase finally into the nitrogenous bases and sugar. Is it clear? But all the enzymes, if you see, they are present on the brush bordered cells of the mucosa. And the mucus along with the bicarbonate from the pancreas. The pancreas is secreting the bicarbonate ion also. Will protect the intestinal mucosa from acid as well as provide alkaline medium for enzyme activity. In the intestine, the pH is 7.8. They will maintain the pH 7.8. Some mucosa gland, Brunner's glands are also helping in making this contribution called succus entericus. So I think so this much topic is clear, beta. So breakdown of the biomacromolecules occur in the duodenum region of small intestine. Simple substances which are formed are absorbed in jejunum and ileum. And undigested material or unabsorbed, which is not digested, which is neither absorbed, is passed on to the large intestine. So in this video, I try to explain you the digestion done in the small intestine with the help of pancreatic juice, how the bile juice are helping in digestion, and end products are digested finally by the intestinal juice. The pH of the intestine is 7.8, which is achieved with the help of the bicarbonate coming from pancreas and the secretion coming from the Brunner's gland, which are present in submucosa. Now let me discuss about the large intestine digestion. Now see beta, no significant digestive activity occur in large intestine, because we have already studied that undigested and unabsorbed are passed into the large intestine. Then what is the function of large intestine? Function is to absorb the water, minerals and certain drugs. Please, this is question area. Then second is it is helping in secretion of mucus, which help in adhering the waste particle and lubricating it for easy passage. Now undigested and unabsorbed substance, which is known as fecus, will enter into the cecum. You can see in this diagram, see here, you focus better here. This is known as ileocacal wall. Between the ileum and cecum, this is known as ileocacal wall. Clear, but a ileocacal sphincter is present. So it is entering into the cecum of the large intestine through ileocacal wall, and which prevents the backflow of the fecal matter. And it is temporarily stored, fecal matter is temporarily stored in rectum till defecation. So I hope so in this video, we have studied today the digestion of the biomacromolecules with the help of pancreatic enzyme, how the bile salts are helping in emulsification, and how the final end product is converted with the help of the intestinal juice by releasing disaccharidase, dipeptidase, lipase, nucleosidase, and aminopeptidase. and no digestion is occurring in the large intestine. I hope this video is helpful for you. Thanks for watching.